Welcome to the course Principles of Palaeoecology, lecture Proxies, first part. In this lecture I will touch on such topics as what is a proxy, types and varieties of proxies, spatial temporal resolution, plant macrofossils, pollen and non-pollen pollinomorphs. All palaeoecological results and interpretations are based on and inferred through the evidence, or as we call proxies. Different sedimentary environment contains different proxies, which can tell about the past living and environmental conditions. In order to study the dynamics of ecosystems, it is necessary to look back into the past by using the record of changes in fossil organisms and sediment characteristics, or proxy data. Because of complex network of interactions through the ecosystem, it is desirable to study as many proxies as possible in order to gain a wider overview of the situation than could be acquired from, let's say, a single proxy. All palaeoecological results and interpretations are based and inferred through the evidence as proxies, as I already mentioned, and there are numerous proxies used in palaeoecology, and therefore only a brief overview of few proxies is given further in this lecture. Proxies first part, and there will be also a second part. So let's begin with the pollen. Although pollen have been studied earlier, it was not until the first proper microscopes appeared to look into microscopic objects. In 1836, Henrik Robert Goppert observed one of the first pollen from sedimentary rocks in Germany. However, the scientific world assumed the birth of modern pollen analysis was in 1916, when Swedish geologist Lennart von Post introduced with the concept of modern pollen analysis. After this year, pollen analysis was applied to a variety of sediment types and places around the world. And even today, we are extensively using pollen analysis for such purposes as vegetation and climate reconstructions. Pollen is a fine to coarse powdery substance comprising pollen grains, which are basically male microgametypes of seed plants. Pollen grains have a hard coat made of sporopollenin that protects them during of their movement to female flower, or as we call stigma, for pollination. Pollen and spores are usually considered together within the palynology. So the palynology is a study of pollen. And pollen grains are plant parts produced in anthers, often in different flowers from ovules. Gymnosperms are a group of seed-producing plants, for instance conifers. Gymnosperm seeds develop either on the surface of scales or leaves, often modified to form cones. Well, in most cases these are cones. Well, the outer part of or walls of pollen grains are made of cellulose and sporopollenin, as I already touched on this topic, which allows their preservation under limited oxygen conditions, or as we call them, anoxic conditions. So these pollen grains can preserve centuries to even billion years. Sporopollenin is usually resistant to most forms of chemical and physical degradation except oxidation. So if the spores and pollen is open for the oxygen environment, then it most likely will go dissolve or go away, degrade. And the living contents of the grain may die within hours or days of release, but the outer wall can survive for considerable periods. And plants have different pollination strategies. So there are Probably you have heard about wind pollination, animal pollination, and self-pollination. On the left side, I put these two gifs for better understanding, and on the left side is an example how much pollen can be emitted from one tree. Pollen gets into the air, and at some point it falls on the ground, soil, in peat, and maybe on the lake surface. In all these environments, pollen grain becomes as a part of sediment. Well, this process occurs every year, so importantly, various plant taxa disperse pollen or spores, which differs, and if they are different by the morphology, it is possible to say which pollen or spore belongs to which plant. This is how we can say which plants were once growing and how did the landscape look like. And when we do so, we call it vegetation reconstruction. So let's zoom in into the pollen grain, and the outermost layer of pollen cell walls, or exine as we call, serves as a robust physical and chemical barrier protecting genetic material. Each plant taxa have their specific pollen types that allow their identification from the sediment and reconstruction of the past vegetation, as I mentioned. So the pollen grain forms this wall consists of exine and intine. Yeah? Well, the latest studies show that plants favor the production of uneven asymmetrical patterns of the surface of pollen grains over more symmetrical patterns. And plants with wind dispersed pollen tend to produce more than those with insect dispersed pollen. In this slide, I would like to stress that pollen grains are usually yellow, brownish, yellowish in color, and that is why they contain carotid elements inside them, and this specific compound helps to then protect against UVB insolation, so basically from the desegregation of due to the sunlight or sun or light waves. Yeah, so pollen appears to need this protection from UVB insolation, and some protection is provided by yellow flavonoids and some other compounds. So this is why they are yellow. Pollen grains are usually spherical or elliptical, and they are different in diameter from about 10 micrometers to 100 micrometers. So 1 micrometer, let's say 10 micrometers is 0.01 millimeter, while 100 micrometers is 0.1 millimeter. And pollen extraction from sediments and preparation method itself have changed little since the von Post introduced it to the scientific world in 1916. Well, in chemical treatment, we use 10% concentrated hydrofluoric acid. Then we use also 10% concentrated strong potassium. And what else? It's like 99% concentrated acetic acid, 99% concentrated acetic anhydride, and of course sulfuric acid. And sometimes to get rid of sand and clay, we treat sample with hydrofluoric acid, HF. And this one is really nasty because it dissolves rocks, and if HF gets on skin, it can burn through the skin and even bones and lead to the death. So there are no jokes about the treatment in the lab. And we can not keep this HF in glass beakers and bottles because glass is made from silica or sand, and HF will simply dissolve it. Hence, we are really careful when treatment with HF is necessary. And there are even labs in the world which even do not allow to use HF due to high risk of danger. Well, nevertheless, pollen is so strong to survive after all of these steps, and that is something remarkable. Our pollen method is one of the most frequently applied methods in palaeoecology and has been in use for nearly a century now, initially as a method for investigating past climatic changes. For example, in 2015, we published an article in the Journal of Palaeoecology where we used pollen to reconstruct mean air temperatures for the eastern Latvia for the last 15,000 years. As you can see in the graph, then on the x-axis there is the time. At the right side of you see the oldest time, and on the left side mark 0 indicates today. So let's add today's winter temperature for Rezekne city, which is the closest point for the sampling point. In this way we can evaluate that there were times when mean winter temperature was lower than present. So interesting point is that and this mean winter temperature today for Rezekne city is about minus 7.5 degrees. So interesting point is that from 8,000 years to 4,000 years ago, there was a time when mean winter air temperature was 2 to even 3 degrees higher than today. Over the last 4,000 years only, temperature gradually decreased until the last century, when it again rapidly rises again, as we now know. About the climate we will talk in different lecture, but this is just a glimpse how we can use pollen analysis for what purposes. So the next palaeoecological proxy, because we talk today about proxy, and this is a wide group of proxy. Well, it's a one proxy, non-pollen polymorphs, but within this proxy we have different other proxies. So let's dive into the non-pollen polymorphs. So it was not until 1968 when Bas van Gaal, at that time a master's student at the University of Amsterdam, started a systematic study of non-pollen polymorphs found from the pollinological slides. So from the pollen analysis, he looked on a pollen, but he recognized that there are something else, not only pollen but also other microscopic objects seen on the slide. So now a standard for pollinologists is to describe the non-pollen polymorph types and to give them a name, even if they are of unknown origin. The list of new non-pollen polymorphs is increasing. However, many of them still remain unidentified. Until 2012, non-pollen polymorphs named as type, number, but now they are renamed by substituting type for HDV, meaning the Hugo de Vries laboratory. So probably my pronunciation is not correct for this, but Hugo de Vries laboratory. So we change type to HDV. And significance of non-pollen polymorphs is growing, as it can be seen also from the Scopus database, where documents related with non-pollen polymorphs increases. So what I did, I just took a glimpse in the Scopus database and type and search for the keywords as non-pollen polymorphs, and this is what I got. So there are two graphs until 2012 and also in today's perspective what we see. Well, 2019 is still ongoing, so I assume the number will documents listing non-pollen polymorphs will be even higher. So based on environment, there are aquatic and terrestrial non-pollen polymorphs. To name few terrestrial examples, I will list leaf stomata, parasitic fungi, symbiotic or mutualistic fungi, coprophilous fungi, pyrophilous fungi, also phytolites. And within the aquatic Non-pollen polymorphs most frequently can be associated with chlorophyta or green algae and other algae, and also cyanobacteria, as well as Nymphaea sclerate, which is water lily sclerate. Well, one example of routine pollen slide counting where in the center is visible parasitic fungi spore of Kreitzmaria deusta. Yeah, so this brown spore is fungal parasite on tilia. And well, it looks like that's it. Nothing else can be seen here. And but vaguely on the right side is also algal remain of Glaucospira. Well, the size of the Kreitzmaria deusta is about 25 microns, and therefore it takes some effort to count all these small non-pollen polymorphs. Yeah, but again, you have this insight into the terrestrial and aquatic ecosystem, which is important. That from one slide you can get information about two environments. Within the non-pollen polymorphs, let's see what is under the algae and cyanobacteria. Well, commonly diatoms and algal pigments have been analyzed in paleolimnological studies, but there are also other phytoplankton representatives such as Stanidesmus, Tetraedron, Pediastrum, Staurastrum, Botryococcus, and cyanobacteria that can be studied in more details. Yeah, I added some few pictures of these remains, how they look like, and well, in today's perspective, algae and the pollution and eutrophication of the water bodies is a huge thing and topic in the worldwide, as well as within the European Union, as in the frame of water directive frame, yeah. And why these remains are so important? Because phytoplankton are, well, they are microscopic organisms, but they inhabit the upper sunlit layer of almost all oceans and bodies of fresh water. They are agents for primary production, so they form a basis for food web, as you can see, from the bottom to the top here. So at first we have these algae, then zooplankton, benthos, and so on, and at the top we have higher organisms as fish in this case. Yeah, so as they are primary producers in the lake, they create organic compounds from carbon dioxide, which is dissolved in the water. Yeah? And phytoplankton obtain energy through the process of the photosynthesis and must therefore live in the well-lit surface layer of an ocean, sea, lake, or other body of water. That means algae and phytoplankton, as it stands for phytoplankton, it needs photosynthesis to live, to live on, and that's why they tend to live on the surface layers. The water lily sclerates. Water lily sclerates appear as isolated cells which differ from neighboring plant tissues by size, shape, and thickness of their wall. Here is one example from pollen slide, what I found from lake sediment. So this is water lily sclerate. And these features provide support for the leaf and stem. Sclerates cause the plant tissue to be coarse and gritty, and suggesting such as adaptation against herbivory. Plants protect them against animals. They want to be living, not to be ate by the animals. So all plants, as we know, have some strategy for protections. Yeah, and stomata is the next one. Stomata can be found in all leaves. They can be found in all leaves, and thanks to which plant breathes. As you can see, through the stomata, carbon dioxide enters and water and oxygen exit. Yeah, and all leaves have these stomata, thanks to which they are able to breathe and make photosynthesis. The stomata themselves are cell structures around the air hole of plant leaves and needles. Especially conifer stomata is preserved in the sediments and reveals the local presence of the tree species. Conifer needles usually do not fall far from their stand, and therefore, if we find a needle of spruce or pine in the sediment, we can be sure that this is the direct evidence of presence of species at the site. As you know, then organic compounds decompose, and then what is left in the sediment are stomata. So let's assume the needle is falling on the ground, but during the time it decomposes, but the leftover is stomata. And crucially, that stomata can be counted from the same slides as pollen. So even such microscopic object can be used as sure evidence for conifer tree presence. Yeah? So stomata, just remember, stomata also can be as a proxy, not only for the presence of the trees, but also to study the climate and CO2 concentration change in the past. The next non-pollen pollinomorphs category and palaeoecological proxy are fungi. These include variety of remains from the fungal vegetative compounds to fungal tissue remains and spores. Fungi form a close relationship with plants for mutual benefit. They are able to get nutrients such as phosphorus and nitrogen out of the soil and exchange them for sugars that the plant makes by photosynthesis. And this process dates back over 400 million years, and approximately 80% of all modern land plants can form this symbiosis, thus showing just how important this friendship is. And there are even numerous studies underlining that without fungi, plants would be weaker and would lack of crucial nutrients, because fungi helps to uptake necessary nutrients, as I already mentioned. Here, the picture on the left side, you see a plant. Well, basically, both plants have been planted at the same time. On the left side, you see a plant which does not contain any mycorrhiza, which is the fungal spore, the fungi added to this pot, while on the right side there is a plant which contains this mycorrhizosphere. So in the lack of mycorrhizosphere, the plants most likely will be weaker, and that is just showing how significant this connection between the fungi is. And as studies show, then fungi strongly participate in weathering processes such as breakage and dissolution of rocks and minerals. So the weathering process in geology means that, let's say you have rock which during the time gets less and less, so it breaks down and dissolves. And there are different weathering types, and one of those is bio-weathering, which means the vegetation and the living organisms and bacteria is making this weathering. And it has been estimated that fungi account for nearly 50% of the overall bio-weathering, which is significantly larger than the previous estimates were indicated. And here I listed only a few fungal spores. So let's start with this picture, which is indicating that there is a plant and there is a section below the ground where roots are growing. And within these roots, several fungi can be associated. Fungal hyphaea is the vegetative stage of fungal growth, and the glomus HDV 207 spores are also formed under the ground within the roots, like also Diporotheca HDV 143, which is indicator of solanum or potato, or other species. Well, solanum is a genus, a larger genus within the different species inside, but one of those is also potato. So if I would find, let's say, Diporotheca, then I could say maybe this specific plant could be growing there at some point. But why I show this slide is because if you sometimes get analyzed lake sediment, then if you find these from the lake sediment, these remains of fungal spores and remains, then you can be sure that there was a soil erosion, because it is no other way how these objects could be transported to the lake. Yeah? And here is also one other which I forgot to show you yet, but here it is. It's an indicator of carex, sage. Yeah? And once more, Kreitzmaria deusta, which is parasitic fungus on tilia. And if to find these spores from the sediment, we can reconstruct interaction between these species and pathogen and try to answer to the question such as, let's say, how soon trees recover after this pathogen attack. This is just one off top of my head. Populus. Yeah. So there is a tree populus which produce few pollen grains, and these usually do not preserve well in the sediment. But fortunately, there is a fungi which forms a mutualistic relationship between populus, and if I find this spore, I can say for sure that populus was growing in the close vicinity. Furthermore, there is Meliola elisi, which is a parasite on heather. And in the case, if I find this spore, I can say that Kaluna was growing there, even if I do not find any Kaluna pollen. An interesting part here is that although Meliola elisi is a parasite, Isthmospora spinosa is a parasite on Meliola elisi, and therefore, by the inference, if there are no heather pollen, no Meliola elisi spores, but there are Isthmospora spinosa remains, I can say that both heather and Meliola was growing there. I mean that if you find only this hyperparasite, you can be sure that also Kaluna was there. After the fire, specific fungal spores can be formed, namely Neurospora, and the ascospores do not germinate under ordinary cultural conditions. So, but they grow readily after having been subjected to moist heat at 65 to 70 degrees for a few minutes, and therefore these spores can be used to reconstruct past fire episodes. So in the nature, you can't get that high temperatures as 65 to 70 degrees elsewhere as only during the fire. In the lab conditions, in the laboratory conditions, these spores have been treated and made some experiments with them, and we now know for sure that these are related or growing after the fire. All the pollen that was not used for reproduction turns to a nutrient as a source of nutrients. Well, pollen serves as a supplementary seasonal source of nutrients, particularly nitrogen, phosphorus, and potassium for litter and wood decaying fungi. Saprotrophic fungi, we name them as saprotrophic fungi. In terms of energy and nutrient balance, the attack of pine pollen is very valuable. So what you see here is the pollen grains, which have been attacked by the fungi. So this is a nature how the, let's say, fungi penetrate the pollen, which at first contains these nutrients, living compounds inside them, but the fungi feels that they have this compound and penetrate through the openings, through the walls, pollen, and sucks out all the necessary nutrients. And then in the sediment I found, now and then I find that pollen grains have these remains of fungi inside still. So I see directly that this process has been occurring at some point in the past. So that means we can also have more insight what's going on at the soil level, how much nutrients were there, and I mean the fungi usually will not attack pollen grains too often, but if we see such episodes, then we can say something more about the ecological environment as such. So pollen rain is therefore also translocation of nutrients. So within the ecosystem, especially in bogs and boreal forests, as it shows Paleo records. Yeah. So when the pollen is emitted, and we don't, as a humans, we still think that this is a waste of energy to emit so vast amounts of pollen grains. But apparently, nature knows what it does, and it emits enormous amount of pollen, which afterwards also settles on the ground and serves as an additional nutrient source. So let's say if the upcoming years are not so well for the plant, then it can ask to fungi to ensure additional nutrients. Yeah. So it's kind of investment for the future as well. Dung or fecal spores, as these are listed here, has also really specific area and of non-pollen polymers, which is widely now expanding. And these spores indicate presence of herbivores, so large animals feeding on the grass or herbivores. Yeah? And thanks to these, we can reconstruct herbivore presence and population densities. In later lectures, we will talk more about non-pollen polymers and how to use such information. But at this point, it is important that you are aware of such spores. And sometimes some scientists even claim that based on certain dung spores, it is possible to say which animals were living there. Like in this case, Bombardioidea indicates of Alces or elk or moose. How do you like pronounce? Yeah. And this is one example. Just to keep in mind, different substrates might have different fungal spores, and not all fungi and non-pollen polymers preserve. So we have information mostly selective information about specific certain organisms and their remains of the past, which can give some additional information. So this is the end of the first lecture, Proxies 1, and in a due time I will prepare and upload the next lecture, Proxies 2, and see you next time.